All right, welcome to your DNA video notes. So to start out these notes, we're going to talk about some of the scientists that have had a contribution to finding the structure of DNA. So first we're going to start with Frederick Griffith. So he was a British scientist in 1928, and he tried to learn how different types of bacteria cause pneumonia. What he did was he isolated two different strands of bacteria that were in mice, and he grew them in the lab. The importance was this was simply just the first major experiment that led to the discovery of genetic information being passed to organisms. He called this process transformation because one strand turned into another strand. Next up, we had Oswald Avery, who was another physician medical researcher. This was in 1931. So he repeated Griffith's work to determine which molecule was most important, and he realized that when S cells were killed, DNA was released. And then DNA went into the R bacteria cell DNA and changed them into S bacteria. So we had S and R cells, to keep that straight. He concluded that DNA was the transforming factor, aka DNA carries the genetic code. After him was the Hershey Chase experiment. So these were actually two people, and one was a bacteriologist, the other was a geneticist. And this was in the 1950s, so we've progressed a few years here. They've studied bacteriophages, which, if you remember, are viruses that infect bacteria. And they wanted to know which part of the bacteria was infected by the virus, whether the protein of the virus infected the bacteria or the DNA of the virus did. So they realized that non-living particles smaller than a cell can infect organisms, and that they have DNA or RNA, and that's what's injected into the bacteria that they are infecting. So it determined ultimately that genes were made of DNA, and they need this information to produce new viruses. Then we had Herman Chargaff. Now, Herman was in 1950 as well, and he was really important to discovering the structure of DNA in terms of the bases. So his results showed that guanine and cytosine are equal, and adenine and thymine is equal. So he found out that A equals T and G equals C, which you will say to yourself a lot as we go through this part of the unit. Next up, Rosalind Franklin. So there's some controversy with her. She was a biophysicist, but also worked with X-rays. And she had a very famous X-ray image that showed the first initial structures of DNA. She wasn't actually given the credit of discovering the structure of DNA, which you'll see, but she did help with that initial discovery because she did see a coil-like structure in her X-ray, aka a helix, which we now know that DNA is a double helix. Finally, we had Watson and Crick. So this was in 1953, and their goal was to construct a model of DNA. So they used everybody previously, they used all of their work, and they formed the DNA double helix molecule model. Now, this was the first model of DNA. It showed a backbone made of sugars and phosphates, and it showed base pairs, so A and T and G and C in equal amounts. So now that we know about the scientists, let's look at this structure. So DNA stands for deoxyribonucleic acid, and it's important that you think of it almost like a ladder. So the rails of the ladder are made of sugar and phosphate. They alternate down the side. And the steps then are base pairs, so A and T, G and C. All three of these things combined equal a nucleotide, and it's really important that you know this. A nucleotide is a sugar plus a phosphate plus a nitrogenous base. You need to highlight this, star this, underline this. This is super, super important in your notes. So here's the nitrogenous bases in terms of their structure. So two of them are double ringed and two of them are single ringed. Now, if you notice, a double ring pairs with a single ring. So a purine and a pyrimidine are always going to pair together. There's not going to be two pyrimidines that pair or two purines that pair. So as you see, we know that A pairs with T and G pairs with C. You also need to remember which ones are which. So cytosine and thymine are pyrimidines. How I remember this, they both have Y's in them, and pyrimidine has a Y in it. So cytosine, thymine are both pyrimidines, and then by default, adenine and guanine are purines. So the bases only pair certain ways because of the base pairing rules. So like we said, purines, pyrimidines pair together, and there's something called hydrogen bonds that hold them together. Now, hydrogen bonds are weak bonds that are easily broken during DNA replication or protein synthesis, which we'll see later. So let's do a quick review of the structure of DNA and then chromosomes. So DNA together forms something called histones. So it's going to coil around these histones. Histones are going to form nucleosomes. So this is how we get our chromatin fibers. So the nucleosomes to the chromatin is eventually going to condense to form chromosomes. So DNA replication is simply the process of making DNA from DNA. So why does DNA even replicate? Well, if you remember, we talked about mitosis. Each cell has to get equal amounts of DNA. So it has to replicate, that way the cells are identical at the end. If you also remember, DNA replication occurs in the S phase of interphase. And as a result, we get two identical but complementary strands. And remember, these are always going to follow the base pairing rules. So how does it replicate? So each strand has all of the information it needs to make more DNA, but it's different depending on the organism. So in prokaryotes, the replication is at a single fork and it continues in both directions until complete. It's usually like an oval shape. Eukaryotes, on the other hand, we have a big cluster of DNA in our bodies and our cells. So the replication begins at hundreds of places and it goes in both directions until everything is replicated. So there's 4 important parts you need to be aware of DNA replication. So we have initiation, unwinding, elongation, termination, and we're going to discuss those in a little more detail here. In initiation, this is when replication begins. So an initiator protein is going to bind to the origin of replication. Remember, in prokaryotes, there's only one of these, but in eukaryotes, there's hundreds of them. Then the unwinding occurs. So before it can even replicate, it has to unwind. Remember, it's in that twisted ladder form, so it has to unwind to where another strand can easily form off of it. So it's held apart by single-stranding binding proteins. These are going to hold the DNA open, and something called DNA helicase unzips the DNA. So this is how those hydrogen bonds are broken that we talked about. So as this happens, there's going to be 2 strands that end up being exposed. So you're going to have a lagging strand and a leading strand. The lagging strand is a 5' to 3', and we'll discuss that more in class. So DNA is going to discontinuously replicate here, and it produces something called Okazaki fragments that are later joined. The leading strand is a 3 to 5 template. So this goes through continuous replication. It doesn't ever have to stop. So in elongation, it's important to remember that replication is always going to move from 5' to 3'. This is important. Make sure you star or highlight that. And like we said, replication begins at the replication fork. Something called DNA primase is going to be on the lagging strand, or I'm sorry, this is not DNA. This should be RNA, excuse me. RNA primase is on the lagging strand, and it adds a short segment of RNA onto the lagging DNA strand. From there, DNA polymerase will be able to add free nucleotides to the chain. And please make sure you correct this in your notes. This is supposed to be RNA primase. So here's a look at this process. So like I said, we have our helicase here, which is unzipping. Then you have lagging strand here, leading strand here. As you can see, these little pink arrows are your Okazaki fragments. And then these little green spots is your RNA primers. So the primers attach first, then this DNA polymerase is going to add free nucleotides, or A, T, G, or C, to the strand to create those Okazaki fragments. Well, DNA ligase is going to move this way, and it's going to connect all of those pieces. And on this side, on the leading strand, as you can see, DNA polymerase starts at the end, goes down, it never has to stop. Here's another look at these 2 processes with leading and lagging. So at termination, how does the DNA know to stop? Well, in some DNA, when 2 forks meet, this is where they stop. In others, there's a specific termination sequence on the DNA that means to stop. And then, like I said, with the DNA ligase, so the Okazaki fragments are rejoined using something called DNA ligase. I think ligase link. It links those fragments together to form a full strand. So here's another summary of it. If I were you, I'd pause this here, and you could either sketch this in your notes or screenshot it. This is going to be important for you to reference later. So here's a review of the process. We have initiation, elongation, termination. So in DNA replication models, there's also some important things you need to know. So there's several models, and you can read through conservative and dispersive, but what you really need to focus on is semi-conservative. Semi-conservative is the DNA replication model that's accepted. So what this means is each strand of DNA serves as a template. So each side is going to make another strand on it. So what happens is we get this. You see our dark blue starts, but then these ones have one dark blue and one light blue. So each one has one of the parent strands and one of the new strands. And then if they keep going, you'll see the same thing. One of the parent, one of the new strands in each model. The semi-conservative, like I said, is the important one that you need to focus on. Don't really worry about the other two. So what about errors? So when we have errors, there's actually only one mistake per billion nucleotides that happens. And they're caught or fixed in several ways. One, nucleotide selection. So they're paired with their complements at a very high accuracy rate, A and T, G and C. This is pretty simple and it's a pretty easy way to prevent errors. Second, though, there is proofreading. So if an incorrect base is added, DNA polymerase is going to stop, remove it, and then replace it and keep going. Finally, mismatch repair. So if number 1 and number 2 fail and you do truly have an error, mismatch repair enzymes are going to go find the problem and they will replace the mismatched base with the correct one. So let's do a little practice of DNA replication before we end here. So basically you're just giving the complementary strand for each. Now I can't write as small as this is typed, so bear with me. So C is going to pair with G, C with G, G with C, T with A, T with A with T, T with A, G with C. Now if I were you, I'd pause it here, try to do the other two, and then push play and check your work. So for the second strand, we have T pairs with A pairs with T, G pairs with C, G pairs with C, C pairs with G, T pairs with A pairs with T, A pairs with T. And finally, we have A pairs with T, G pairs with C, C pairs with G, C pairs with G, C pairs with G, T pairs with A pairs with T, and G pairs with C. And that's all for your notes.